Hi everyone! Ever wondered how scientists sniff out cancer causing chemicals choosing just bacteria? Welcome to the world of Ames Test, where tiny microbes play detective in the fight against mutagens. Hi, I am Nabanita, a cancer biologist and science educator. Today, we are diving into one of the coolest, simplest, and most powerful tools in molecular toxicology, the Ames test. Meet Salmonella typhimurium. These bacteria have a mutation that stops them from making histidine, an essential amino acid. So, they can't grow unless we give them histidine. Now, here's the twist. If we expose this bacteria to a chemical that causes mutations, some of them might mutate back, regaining the ability to make histidine. And boom, they grow. The more colonies we see, the more mutations have occurred. And that tells us this chemical might be a mutagen, possibly even a carcinogen. From cigarette smoke to food additives, the Ames test has flagged countless compounds that could damage our DNA. It's fast, cost-effective, and surprisingly elegant. We can add liver enzymes to mimic metabolism because some chemicals only become dangerous after our body processes them. The test was developed by Dr. Bruce Ames, an American biochemist and molecular biologist. He was driven by the need of a simple, rapid, and inexpensive method to screen chemicals for carcinogenic potential. His work revolutionized toxicology and environmental safety testing. For all the potential drug candidates, this test is a must. Now come to the protocol of this test. First, we have to prepare the mutant bacterial strains for the test. We have to use the strains of Salmonella typhimurium which carry a mutation in genes required for histidine synthesis. Now these strains cannot grow on media which lack histidine unless a reverse mutation occurs. So first we have to spread these mutant bacteria on agar plates that lack histidine. Then we have to include a small amount of histidine to allow at least a few cell divisions so that in future these cells can serve as the mother cells for mutations to occur. Now we have to apply the chemical which we wish to test. We have to apply that chemical on the plate. Usually we also add S9 liver extract from rats to simulate mammalian metabolism because some compounds become mutagenic or carcinogenic only after metabolic activation. And then the incubation period. We have to incubate those plates at 37 degrees centigrade for 48 to 72 hours. If the test chemical is not a mutagen, if it is safe, we cannot see colonies of Salmonella typhimurium in the plates. But if the chemical is a mutagen, it will induce reverse mutations in some of those mother cells. These mutations will restore the ability to synthesize histidine in those mother cells. So colonies will grow on the histidine-free medium, and then we have to compare the number of colonies to the control plate where no chemical was added. So what is the significance of this test? If we can see a significant increase in the relevant colonies in the agar plates, that means the chemical is mutagenic. So next time you hear about chemical safety testing, remember a humble bacterium might have been the first to raise the alarm. If you found this fascinating, hit like, subscribe, and drop your questions in the comments. Science is cooler when we explore it together. Thanks for watching and bye.